Good evening, all. This is actually the second talk of the ongoing Samvaad series on medical AI. And this is also the first or our first guest talk of the year. So today we have with us Professor Anubha Gupta from IIIT Delhi as our guest speaker. Professor Dinesh Babu Jayagopi is the session chair for today's talk. Professor Dinesh is currently an associate professor here at IIIT-Bangalore, and he heads the multimodal perception lab. And he's also one of our key persons to go for signal processing and ML related things. So without further delay, let me actually hand over the stage to Professor Dinesh. Over to you, Professor Dinesh.
Yeah. Thank you, Nikhil, and welcome, Professor Anubha Gupta. We'll do a quick introduction. Professor Anubha Gupta received her BTech in 1991 and MTech in 1997 from Delhi University in ECE. She received her PhD from IIT Delhi in electrical engineering in 2006. She did her second master's as a full-time student from University of Maryland, College Park, USA, 2008 to 2010 in education. She worked as assistant director with the Ministry of Information and Broadcasting, Government of India through IES from 1993 to 1999, and as faculty at NSUT Delhi 2000 to 2008 and IIIT Hyderabad 2011 to 2013. Currently, she is working as professor at IIIT Delhi. She convenes the Signal Processing and Bio Imaging Lab, SBILab, and she has co-authored more than 100 technical papers in scientific journals and conferences in both engineering and education. Her research interests include applications of machine learning in cancer genomics, cancer imaging, biomedical signal and image processing, including fMRI, MRI, EEG, ECG, signal processing, and wavelets in deep learning. Dr. Gupta is a senior member of IEEE Signal Processing Society, SPS, and a member of IEEE Women in Engineering Society. She is an associate editor of IEEE Access Journal, IEEE SPS Magazine newsletter, e-newsletter, Frontiers in Neuroscience, and IET Journal of Research. She is also the technical committee member of BISP, which is Bioimaging and Signal Processing Committee of IEEE SPS Society for Jan 2022 to December 2024. Welcome, Professor Anubha. Over to you.
Yeah, thank you, Professor Dinesh. So my talk is titled AI in Blood Cancer Imaging. So in this talk, I'll be speaking about all the work that has been done in my lab in the area of blood cancer imaging, largely in the diagnostic area. So before starting with the technical session, so I would like to briefly outline the current research work that we are taking up in the SBILab. So we are largely working in the area of medical image analysis, particularly cancer image processing, segmentation of cells, classification of healthy versus cancer cells. In the area of cancer genomics, where we are working with next generation sequencing data, cancer survival analysis. All these works are being done in blood cancer area. Then we are also collaborating with some local hospitals in Delhi and working on cardiovascular disorders, particularly using ECG signal processing. We are also working on EEG and MEG signal processing, and of course fMRI, DTI, etc. For this work, which is on AI in blood cancer imaging, we have run 2 funded projects, which were led by myself and my collaborator, Professor Ritu Gupta, who is also heading the laboratory oncology unit at AIIMS, New Delhi. A large part of the work that I'm going to present has been done by my PhD student, Shiv Gehlot. But a large number of research staff, interns, and students have contributed over years in this work. So I acknowledge the support of everyone whosoever has contributed, in particular my student Shubham, Rahul, Pramit, Shruti, Manya, Dhruv, Suril, and many more. So the outline of the presentation goes as follows. I'll be giving a brief introduction about the problem statement, then the stages that we need to build for an automated tool for cancer detection. Again, I'm repeating, it is for blood cancer detection. The challenges we have in hand, the deep learning based solution that we have proposed over time, conclusion and references. So the introduction of the study goes as follows, that we aim to design and develop a computer-assisted tool for blood cancer detection. Now, there are different types of blood cancers. So in this work over these past years, we focused on only two types of blood cancers. One is BLL and the second is the multiple myeloma. Now, both these cancers are because of the malignancy that happens in the white blood cells. So multiple myeloma is because of the plasma cells, a type of white blood cell, and B lineage acute lymphoblastic leukemia is because of the B type of cells, that is B lymphocytes. Now, the significance of the study is that initially, let's say a doctor suspects that a person is suffering from blood cancer. The technique which is used in this particular case is microscopic image analysis, where maybe the peripheral blood is taken and a slide is prepared and is seen under the microscope. Now, if the person is suffering with a particular blood cancer, one may find a large number of cells of a particular type. And that gives the idea to the doctors that the person is suffering with a particular type of cancer. However, when the chemo is administered, the number of cells of a particular type, they reduce substantially, but there may still be a large number of cells in the human body. So in this particular case, when the pathologist makes a slide and looks at it under the microscope, they may not see a huge number of cells of one particular type to detect it. So there comes the challenge in cancer, particularly blood cancer detection after chemotherapy, because the number of cells which could still multiply, cancer cells which could still multiply and again lead to full-blown cancer may be really large, although they are not that large when it comes to looking in the peripheral blood. So microscopy cannot be used, and doctors use the advanced method, which is the flow cytometry. Another challenge that also happens with looking at the cells under the microscope is that morphologically both the healthy and the cancer cells, they look similar. So the doctors cannot, or pathologists cannot differentiate whether it is a cancer cell or the healthy cell. The only way of differentiation under the microscopy image analysis is looking at the number of cells that are present at that time in the blood. So since cancer is because of the unrestricted growth of a particular cell type, and hence if the number of cells of a particular cell increase beyond a certain limit, then they are able to see that it is a cancer, but not because of the differentiation with the eye on whether it is a healthy cell or a cancer cell. So that means that once the number of cells decline, they have to resort to the advanced method. So one of the advanced methods is, for example, flow cytometry. But flow cytometry is a costly equipment. So the infrastructure is costly. Also, it requires a reagent, which also costs a lot, and is run by a skilled manpower. So that leads to prohibitive expenses when it comes to flow cytometry. So the idea was that if we can make an automated tool that can actually work on the bone marrow aspirate fluid and based on the image processing, if it can distinguish that a particular cell is a healthy cell or a cancer cell, then we can count the percentage of cancer cells after the chemotherapy has been administered, and maybe instead of using flow cytometry, based on the simple microscopic plus image processing analysis, we can do the cancer detection. So this would imply that the proposed tool, because it is a computer-based tool, so it can be deployed in urban or the rural areas with ease and wouldn't require, wouldn't run with exorbitant costs in running the flow cytometry tests. So what are the different stages of building such an automated tool? So definitely the first step would be recruitment of subjects and capturing microscopic images from healthy and cancer subjects. Now, I'll also present the slides and show you that over time when you capture the images, we do see that there is a stain color variation from one set of images to another set of images. So stain normalization then becomes the second important step that we needed to take care of. Then the cells can be occurring as a single isolated cell, or they could appear as clusters of cells. So since we are eventually interested in figuring out the percentage of cancer versus healthy cells, so that implies that we would also like to break a cluster of cells, because this could be maybe there is one healthy cell clustering with 3 cancer cells. So hence, the classification has to happen at the cell instance level, and hence we needed to segment the cells from the clusters of cells from the images. And finally, building a classifier, which should be a GUI-based automated tool. So each step of the workflow pipeline has some challenges that we encountered over time. So I'll present now. So for example, if we look at the stain color variations, so we find that, so these are the 3 images which have been captured from the person suffering from BALL cancer, that is the acute lymphoblastic leukemia. Now, if you look at the darker shaded cells, these are the cells of interest. So in all the 3 images, but if you see the 3 images, they have very different color contrast. So actually these images have been collected over a period of 2 years. So during this time, we find that maybe the chemical with which we are staining the slides, that changes, the bottle finishes and another batch of the chemical is received. So with the change of the chemical, or with the change of the camera settings or the microscope settings, or with the change in the staining also, because maybe earlier some other technician was staining and now some other person is staining, although there are set protocols, but still the time during which the stain chemical is put, that may slightly impact the color contrast. So we find that owing to different reasons, the color contrast of the images changes. Now, since the cells of interest, when we are going to segment them and classify them, so these segmentation and the classification pipelines may depend on these color contrast of these cells, and hence it is important to take care of the stain color variations. So these are the 3 images that is corresponding to the multiple myeloma cancer, and again you can see that there is a different color contrast. Even the nucleus looks slightly different in the 2 images or in the 3 images based on what the color contrast is at that time. So, what are the challenges with the segmentation? So which is the second stage of this automated tool. So we find that, for example, this particular image is is captured from the multiple myeloma patient. So these are the 2 plasma cells. So if you look at these plasma cells, and this is the 3rd cell, so if you look at the cells, they may be touching each other. Now there could be 3 scenarios. Maybe the nuclei of the 2 cells are touching each other, because we would also like to cluster, to segment these out of the cluster. So the segmentation then becomes a challenge when maybe the cytoplasm of the 2 cells are touching each other, because the boundary of the 2 cells may be too feeble. The other challenge could be that the cytoplasm, in some of the images, we find that the cytoplasm is actually very near in the color contrast to the background. So that also poses another challenge in segmenting the cytoplasm out. So largely the nucleus segmentation is still relatively easy problem. But when it comes to segmenting the cytoplasm also, then it becomes a bit of a tricky problem. Then if we look at this image, then we find that in this particular cell, the nucleus and the cytoplasm, they also have a similar sort of a shade, although they are still different, but their shade is very near to each other. So, all these sorts of scenarios, they actually pose problem in building a robust segmentation pipeline. Besides these, we also find that there are some stained cells, but they are the unwanted cells. For example, this one, this is not a plasma cell of interest, but it is an unwanted cell which has also got stained. So, this implied that when we are segmenting the cells out, we also needed a marker on which cells are actually the cells of interest. We may see many cells which are actually have got stained. So for example, these cells are the unstained cells which have not got stained when a stain color is, when staining has been done. But all these cells have actually got stained, but not every cell which has got stained is a cell of interest. So here we took the help of the pathologist, where we have built a semi-automated tool where they first put the markers on the cells that they feel are the cells of interest that should be segmented out. And then the automated pipeline should segment the cells out. So if you look at the sample images, for example, on the top row, we have the nuclei which have been segmented out of the cancer cells. And then at the bottom, we see nuclei that have been segmented of the cells of the healthy subjects. So looking at these images or looking at these sample nuclei, again, we see that it's not possible to infer which cell is healthy cell and which cell is cancer cell. So both the rows almost sort of look alike. And hence it is difficult to just by looking under the microscope, one cannot annotate which cells are the cells of interest, which cells are the cancer cells and which ones are the healthy cells. So what are the challenges with the classification pipeline or the classification step, which is the final step of this entire tool? So the first one is there is a visual similarity in the samples of the two classes. Now, the second challenge that we also faced was that the data set was, at least BLL data set that we built initially, was actually highly imbalanced. And why it happens is that, for example, how we collected the data was that we collected healthy cells from the subjects who were not suffering with any cancer. And we collected cancer cells from the subjects who were suffering with BLL cancer or cancer, and they were at that diagnosed and they had full-blown cancer. That is, they were not administered any therapy at that time. So what we found that in the microscopic images, when it is a healthy subject, then because the number of cells are very less, so we could segment only few cell images out of the healthy subjects. Whereas when it comes to the cancer subject, then in every slide we had a huge number of cells of that type, and hence the number of cell images that we could collect from the cancer subjects was very large as compared to this, the number of cells that we collected from the healthy subject. So that led to a ratio of roughly about 2:1, twice the number of cancer cells were there in the data set as compared to the healthy subject. So this would often happen that the data set is imbalanced. Then we also learned over a period of time that subject level variability plays an important role. Now, this is particularly, I would like to emphasize on this aspect because over time, while working with different medical imaging or medical data sets like ECG or EEG, etc., we know that there is a subject sort of information which is also present in the data. So I'll share, after this, I'll share the first model that we built. And we found that from one subject to another subject, there is a huge variation in the types of images. But at the same time, the subject level information is also important there. Now, what would happen with the medical tool is that once you build the tool, you would like to deploy it in the real-time settings in a hospital. So whether you collect data of maybe, let's say, 100 subjects or 200 subjects, but eventually it would be tested out on a huge number of subjects. So we found that the subject level variability also plays a critical role here in building a robust tool. And we then prepared the data at the subject level. That I'll come to this part. Then both these data sets have a typical thing that there could be a label noise in one of the class, which is actually the cancer class. So as I said, that under the microscope, it is not possible for even the pathologist to figure out whether it is a healthy cell or the cancer cell. So we prepared the ground truth by collecting healthy cells from the subjects who were not suffering with the cancer, and cancer cells from the subjects who were suffering with cancer and were not yet started with the treatment. So we assume that in the cancer class, all the cells that we have captured from one subject, so for example, all the plasma cells that we segmented out of the data of a cancer subject, all those cells are actually cancer in nature. But it could be possible that out of 100 cells, maybe one cell is healthy and 99 cells may be cancer in nature. So there can be label noise in one class, which is the cancer class of this data. So far, if anyone wants to use that, we have released 4 data sets addressing different problems. So for example, the first data set that contains images of both BLN and ML. And this is for doing stain color normalization. So there are very good quality 30 images of each data set with huge variation across the images. So both the data sets we collected over a period of 2 years initially for BLN, and again we took 2 years to collect the MM data set. Then the second data set is about the segmentation of plasma cells, which is a difficult problem because in BLL, it is largely only the nuclei, but in MM data set, it is also the nucleus plus the cytoplasm, which is of very substantial proportion to the cell. Then we also had the second data set we released on this segmentation of plasma cells. This was a larger data set, the third data set. It is also available at Kaggle as well as IIIT Dataport. And then the last data set, which is actually on the cell classification problem, and it contains about 15,114 curated cell images of very good quality of cancer and healthy cells of BLL cancer. So we also ran 2 medical imaging challenges. So just to give a brief, because we are still maintaining the leaderboards of these challenges. So this we conducted in IIIT-B. That was on the classification task on the last data set that I just mentioned. And these were the top entries on the challenge leaderboard. So the weighted F1 score was roughly around 91% here. But later on, we also had we also developed our own in-house models, which have a way higher weighted F1 score than compared to this. So this is another challenge that we ran last year that was on segmentation of plasma cells. And again, the mean IOU, that is intersection over union. So mean IOU, the highest value is so far 93.89%. But you can see that, so all these models, largely these models were ensemble of very deeper architectures. So one may be interested in building a smaller customized architecture with better or similar performance on these data sets. So we come back to the stages for this automated tool for cancer detection. So we captured the data using two different microscopes. So the first one was Nikon, and we collected at 1000x magnification. And the quality of the images, if you see that the quality of image, we captured the raw images of 2560 into 1920 pixels. So roughly about 14 MB was the size of one single image. Then later on, we also collected data from Olympus camera, so which is at 9.4 MB each size of each image. So for example, these images are actually captured from at these resolutions. So one cell here would be roughly around would be contained in a box about 300 cross 300 pixels length. So these are very good quality images, and the cell images that have been released are also very good quality. So this is the stain normalization pipeline that we built in-house. Reference is given here. So it consisted of handling three problems of stain color normalization. That is, the first one is the illumination correction. The second one was the stain color vector correction, where we made use of SVD, that is singular value decomposition, followed by alignment of the bases. And the third was the stain quantity correction. So if you look at the first image, that is the reference image, and you look at these two images on the top row, they are the query 1 and query 2 images. So for example, particularly query 2 image is actually far off from the reference image. But once they are stain color normalized with our proposed method, you can see that the bottom one is the stain color normalized images, and they are very close to the reference image. So we attempted segmentation using in two ways. One was that we use the multi-phase level set approach to do the plasma cell segmentation. So the approach worked fine on a few images, but over time when we collected data of about over 2 years when we collected the data, so the data or the images were also not of very, like all the images were not of very good quality, because there are portions in the slides or portion where the quality of the cells is not that good, and we found that the method didn't work that robustly. So deep learning could be employed. So the first work that we did on cancer versus the cell detection on multiple myeloma was where we segmented only the nuclei out. So we made use of only the nuclei, but definitely in the near future, we plan to build a tool that works on both cyto and nucleus, and we are hoping that that would lead to a much better performance as compared to the tool that we have built on only the nuclei classification. So how did we do the cancer versus normal cell classification using CNN architectures? So I'll be discussing three different architectures here on this classification problem. So this was the first work that we did, and this was called as a stain deconvolutional layer for CNN in medical microscopic imaging. So this work was published and presented in MITI. So the idea was simple, so initially what we have is the RGB color images, and let us say we have segmented the cells out. Now, what we want to do is that we would like to classify whether this cell is a healthy cell or a cancer cell. Now, one approach is that we take the cell image in the RGB space, feed it to a CNN or design a custom designed CNN, and then do the classification. Now, over time, because we were also dealing with the problem of stain color normalization, so we thought to look at the like where the texture, because once we say that we would like to classify, so maybe based on the texture that we want to see whether it is a healthy cell or a cancer cell. So we wanted to see where, in what space the texture would be making sense. So initially, so we look at how the image is formed. So there is a Beer-Lambert law that is used there, and we call it as the optical density space. So the idea is that you look at how the image is formed, that is I, which is in the RGB space, but it's actually formed because of the interaction of the stain colors and the quantity that is absorbed of each of the stain color. So for example, here in all these images, we use the Jenner-Ginsa stain. So we said that, let us say that the stain could be decomposed into 3 constituents. Now, if there are 3 different constituents of the stain, because the cytoplasm of the cell is captured differently with a different color, the nucleus is captured with a different color, and the background is captured with a different color. So if we look at this particular thing, so this is how the image is formed. So where O is called as the optical density space, that is, this particular term is called as a representation of the image in the optical density space. So we could apply this negative of logarithmic function here, where IOC denotes the max background value. So which one can assume as 255 if it is a 0 to 255 levels of pixel values are there. So then this O image can actually be decomposed using SVD as quantity, which contains the quantity of each stain component absorbed at a particular pixel position, and S, which contains the stain color vectors. So if we look at the quantity, this is where actually the texture would play a role. So we came up with the idea, then how about appending a front-end layer to a CNN architecture where we do this sort of a processing and we initialize these stain color vectors, or we build them as the filters, and we initialize them with the reference image stain color vectors, and later on we start doing the fine tuning of that stain color vector also. But eventually, if we implement this particular equation, then we would be having a quantity image where quantity would be corresponding to each of the stain which is absorbed at every pixel position, which is giving the quantification of the texture, where the texture depends on the intracellular components which are present in the cell and could differ from whether it is a nucleus or the cyto or different regions of the images. So this is how we implemented that. We looked at this S inverse and we made 3 kernels out of this. 1 cross 3 kernels, which were fused in the front end layer of the architecture. So this is the initial RGB image. Then we took the negative log of this quantity, and then we had these 3 kernels. And whatever is the output, that is then fed to the, after the nonlinear activation is fed to the CNN model. So this was the proposed stain deconvolutional layer architecture that we proposed. And we found that compared to LXNet, when we are using the LXNet with the SD layer, so that there was an increase in the performance, whether we used LXNet or whether we used texture CNN, where texture CNN implies that what we did was that the last layer of the LXNet, we did the global average pool of every feature map, and that was the texture CNN because it reduces the number of parameters to be learned in the network considerably. So we found that both these architectures along with the SD layer gave relatively better performance. So these are the visual results where the first row corresponds to the cancer cell and the second row corresponds to the healthy cell. So if you look at the first one is the cancer cell, healthy cell, these are the deconvolved images obtained using SVD. And these are the images that are obtained after the fine tuning of the entire architecture was done. So we do find that there is a slight difference in the cancer cell and the healthy cell when it comes to maybe deconvolving these images. Instead of using the RGB, if we use the deconvolved image to the architecture for training it to learn to classify cancer or healthy cell. So this was then further, but however, we made one mistake during this time. This was the earliest work, that we had actually collated all the cancer cells together and all the healthy cells together, irrespective of the subject identifying information. So later on, when we were testing this tool and we figured out that on some of the subjects, it was performing very good, but on some of the subjects, it was not doing a good job. Then we learned that maybe we needed to separate the data at the subject level, in the sense that when it comes to building the train plus validation data set, the val dataset shouldn't contain any single cell of any subject that has already been used in the training dataset. That is, the subject should remain, it should become absolutely separate when it comes to the training set and the test set or the val set, so that it performs relatively good when it is used on the prospective subjects in the hospital setting. So during this time of the second architecture, then by that time we had again segmented and prepared the data set, this time at the subject level. For each subject, we had kept all the images separate, and this is how the data was also released at the TCIA portal on CNMC dataset. So this is the description of the data set. So if you look at the training data has the data of about 101 subjects and contains about 12,000 plus images in the data set. So the number of subjects, you see that the number of subjects vary in different folds. So we made 7-fold of the training data, and the number of subjects in each fold differ, but we kept the number of subjects in such a fashion that the total number of images, the ratio of the total number of images, they remain almost the same in each of the fold. Because every subject, maybe one subject gave us 250 images, cell images, whereas from another subject, we captured maybe only 89 images and so on. So the test data, it consists of 9 cancer subjects and 8 healthy subjects. And the total number of cell images in the test data is 2000 plus, 2586. So this was the whole setup. So we made use of the cross-fold validation. We built 7 different classifiers, did the cross-fold validation, built a majority poll classifier in the end, and then tested them on the cancer versus healthy subjects. So we used a custom design architecture this time instead of LXNet, which also has a huge number of parameters. So we designed a base network, which is given in this reference article. And so we had a base network, which was consisting of very less number of parameters as compared to other. And it had only 6 number of comp layers. Followed by a bilinear pooling. So we have a nuclei of the image, cell image, which is coming in, pass through this network. In the end, we have bilinear pooling layer, and then it is being classified with there is a fully connected layer, and then it is being classified as healthy or cancer cell. So this was the base architecture that we built, and we called it as the SDNet architecture. So we also looked at the impact of the DCT layer. So the idea was inspired from the fact that flow cytometry uses the spectroscopy. So we thought that how about looking at the architecture, how about looking at the DCT layer and fusing somehow this DCT based layer, that is the kernels of this layer are actually nothing but the eigenvectors of, or the basis vectors of the DCT. So how about using this DCT layer and seeing its impact, whether it leads to any increased performance or not. So we did it in 2 ways, that what if we use on the RGB image itself, we use the DCT layer, or what if we use the DCT layer after staying deconvolutional layer. So we found that if we use DCT layer at the front end, we found that this is the healthy cell and this is the cancer cell. We found that the spectrum was largely similar. However, when we used DCT layer after the SD layer, that is the same deconvolutional layer, we found that there is a difference in the spectra of the two, and that could lead to improved performance in the architecture. Now, this again established the utility of the stained deconvolutional layer, as well as we also saw improved performance with the DCT layer. So this is another architecture which we call as a SDCT architecture. So here we have first the SD layer, then DCT layer, and then this base network. And this is how, and these are the different parameters of this. And we trained this architecture. Now, then we came up with another idea that how about still we wanted to push the accuracy further, and we came up with another idea that how about using one auxiliary classifier along with the main classifier, but somehow training this classifier in a different fashion, and maybe using different sort of transformation at the end of the pond layers to capture different characteristics of the features. So we had this was the feature learning subnetwork was nothing but the CNN architecture, but we used two different types of transformation at the end of it, bilinear pooling and spectral averaging. That is, across the maps, we took the average. So at every pixel position. So these are the two different feature maps that came out, and we had the neural network based. So the top one is actually the SDCT architecture, but the bottom one is the architecture which is corresponding to the auxiliary classifier. So how did we train this architecture? So if you look at the first above this dotted line, this is the architecture which is of the SDCT architecture. But the bottom one is the training of the auxiliary classifier. So what we did was that because we also felt that there could be a label noise and that could be playing a key role here. So what we did was that all the samples which were classified correctly during the training, they were actually fed, their feature maps were, their features were taken and passed through this feature maps were taken and passed through the spectral averaging layer to build a feature vector, and then were used to train an auxiliary classifier. So in this particular case, we had used SVM classifier. So I'm repeating. So all the samples which were correctly detected or correctly predicted during the training of the classifier, only those samples were used to actually train this auxiliary classifier. And later on, when we started doing the testing, then we said that we look at the probability with which it is predicting the class of a particular sample. So if, so let us say we fix the tau as to be 0.9. That is, we really want with high probability, the top classifier should predict the sample. So if the probability is lower than this, then we said that maybe we cannot rely on the decision of this classifier, and then we took the decision out of the auxiliary classifier. So this is how we did the testing. So that is why we said that, and we iterated and we empirically figured out what value of theta works for us. So later on, we took somewhere around 0.9 to 0.95. It was really working good. So in the nutshell, so this is the architecture where we have, if it is theta, that is theta is between, between 0.9 to 0.95, whatever we have fixed. So if it is theta, then it is making use of both the top architecture as well as auxiliary classifier to do the detection. Whereas if theta is equal to 1, so that means we are only basing our decision on the lower architecture, that is auxiliary. And when theta is equal to 0, so that means every sample, whatever way it is being predicted from the top classifier, primary classifier, we are going to choose that value. So we say that this is how the two architectures are building. So we found that our proposed architecture, actually the performance of the SDCT architecture was near the CNMC dashboard, which was weighted F1 score was 0.9. But with SDCT auxiliary architecture, this auxiliary classifier, it suddenly improved the performance by 4%, which was a huge jump. And these were also the accuracies that we looked at for each of the subjects in the test data. And you find, because the number of cancer cells in this data set is almost double that of the healthy cells. So we do find that the error is actually lower for the cancer class as compared to the healthy class. So which gives the idea that perhaps a better architecture can definitely be made and the performance can still be pushed further. So this was the work that we did on the BLL dataset. So that was what we built initially. Then later on, we built the dataset of the multiple myeloma. And here we actually paid closer attention to two problems. One, that in this dataset, we didn't want that imbalance to be that large. And the second one was that we wanted to handle the label noise inbuilt into the architecture in a better fashion. So this data set is corresponding to multiple myeloma. We haven't released this data set yet. And if you look at the number of images here, the number of images is also very large. So roughly around 75,000 cell images as compared to around 15,000 cell images that we collected for the BLL cancer. And here, if you look at the cancer versus healthy class, you will find that the total number of cell images of each of the classes, there is not much imbalance. Rather, the two classes are actually, the number of cells in the two classes are in the balanced mode. So in both the training set as well as the test set. So we utilized more number of images we kept more number of images in the test set and utilized relatively less number of images in the training set. And this time we wanted to actually handle the label noise with better clarity as compared to the previous architecture. So there were two different contributions. One, that we proposed a projection loss based classifier architecture to handle to improve the performance along with the cross entropy loss. And then we again came up with a dual branch architecture, and we handled the noisy label in three ways. One was with the sample discarding. Second was with label flipping, if we have found during the training that the label is noisy. And three, using a coupling classifier with sample discarding or label flipping. Now, again, this is important to note that per se, we do not know which of the cancer cells are actually noisy in nature. So that label is still not known. So the architecture has to somehow predict that it is perhaps this particular sample image may have a noisy label. So first of all, we used projection loss, concept of projection loss. So what we did was that we had the input sample imaging coming into a CNN layer. A number of CNN layers are here, CNN this architecture. And then this is the activation, which is at the flattened layer. And we projected this into 3 class vectors. So, or if it is a 3-class classification, and if it is a 2-class classification, so we'll have 2 class vectors C0 and C1. So now our loss actually consists of 2 different losses. So one is, so if we say, talk about only the projection loss, then projection loss consists of this term, that is the probability of the ith label, which is computed like this, and where the distance d is actually between the activation and the and its inner product with the class vector. So if a particular sample belongs to class C0, we would find that this value would be close to 1, and hence the distance would be 0. So if a sample belongs to class 0, that is, then the activation with the inner product of activation vector and the feature, the class vector would give us a minimal distance, and which is what is going in this particular computation of the probability. In addition, we also imposed a constraint that we wanted these class vectors to be normalized, and hence we had the second term. Now this was the projection loss, but we also made use of the cross entropy loss as we come to the next slide. So how we computed these class vectors and the weights of the architecture is by we updated these class vectors as well as the weight vector over time while training the architecture. So this is again a custom design architecture which consists of this time 10 layers, and we have we gave the output to two different layers, two fully connected layers. So one is the simple flattening layers and one is the global averaging pool layer. So here in the upper one, we are using the projection loss, and the lower one, we are making use of the binary cross entropy loss. So this we call as the projection loss with cross entropy loss. So PRLCE net architecture, where we have both these two losses coming into play. So now with this sort of an architecture, we handled the label noise or figured out whether a sample has a noisy label or not. So if both the branches, that is the upper one, the decision of upper one and the lower one are identical, that is YK cap. Prediction of both branches are same, so that means the label is correct and we keep the decision as such. Now, if the two branches are predicting the labels, but both branches are predicting a different class, but both branches are predicting different class with a probability which is greater than some predefined threshold. So that means that both branches are very, very confident, and both branches are predicting the label with a higher confidence. So then there is a problem. So that means this particular sample could have a label noise. Now the third one is that both branches are actually predicting giving different predictions, but their predictions are below a certain threshold. So when it is S2, case S2, in that case, whichever branch is predicting with a higher probability, we keep the prediction as that. But when it is actually S3, then we felt that maybe this time this sample has a noisy label. So we either did sample discarding or label flipping, or made use of, that is, flipped the label of this sample and changed it in the main data set as well, or used a coupling classifier. Now this coupling classifier, the idea of this coupling classifier was similar to SDCT Oxnet theta. That is, wherever both the branches are predicting the same with a higher confidence, so the labels are correct, and only those correct labels are actually fed to train these coupling classifier. And later on, again, we use a threshold whether we are going to take the decision from the primary or from the secondary one. So these are the results that we obtained. So we looked at the weighted F1 score and the balanced accuracy, and we did the ablation study where we had the base cross entropy net architecture without any PRL branch, with the projection loss branch, but only one base architecture was trained. Here, the PRLC complete architecture was trained, but the decision was taken only from the cross entropy branch. These are the results on the test images. So then with the coupling classifier and with different scenarios like label flipping, with sample discarding, with coupling classifier, and so on. So we found that in the end, the architecture, that is the PRLCE architecture with sample discarding and the coupling classifier, actually gave us a very good performance. We used the majority vote and we found that the weighted F1 score as well as the balanced accuracy, they were relatively very high. Then we compared these architectures with the standard architectures of, we also compared with SDCT Oxnet, and we found that its performance was also not very good on these images, which was actually an indicator that perhaps different architecture, this architecture really did a better job as compared to the previous one. Then we also looked at the performance with ShuffleNet, GoogleNet, DenseNet, different state-of-the-art architectures, and we found that our inference time was also lower. Our number of parameters were also very less because we used the custom design architecture, and our performance was also very, very good on these set of images. So in the end, in the conclusion, we proposed a CNN-based unified framework for the diagnosis of multiple myeloma. The problem is indeed challenging because under the microscope, you cannot say which cell belongs to which class. And the method involved using cross entropy loss, projection loss, label noise handling, and a coupling classifier. And we really found very good performance on the at the subject level. So these were the publications. So I hope I'm in time, and different publications that have come out of this work. And in the end, definitely we would like to thank the Department of DST and Multi both for giving us funding for these projects. The first one was on VLL from MIT, and the second one was on multiple myeloma through DST funding. IBM had provided us support for IIII's 2019 challenge, and Springer was kind enough to publish our challenge proceedings. So we really thank all these agencies, as well as all the students who have worked on this problem, interns, research assistants who have worked on this problem. So thank you. I'm open to questions.
Thank you, Professor Anubha. It was a wonderful talk, and in fact, very impactful work with contributions in terms of data, modeling, system, and a successful collaboration as well. Yeah, thank you very much for the talk. And any questions, you could probably type it either in the Zoom or in the YouTube, wherever you are. So there are a couple of questions which have come in the chat window. So one is from Neelam, maybe that is related to the recent work maybe. So maybe we'll take that one first and then take the next question from Ashok Narayanan. So Neelam is asking, is there provision for uncertain class?
So means to say that it doesn't belong to either cancer or healthy. Yeah. So very interesting question. So this is how I would put it. Definitely you can incorporate another class which is uncertain class, but not for cancer versus healthy, but maybe saying, let us say you're building a tool for multiple myeloma, but by mistake, some of the stem cells, they are not actually the plasma cells. In that case, your inference would also be wrong because it's not a plasma cell. So you shouldn't be classifying it as healthy or cancer in order to detect multiple myeloma. So in order to take care of this sort of a noise or the label noise, I would say it's a different type of label noise. In that case, one should make a provision for uncertain class and maybe build a 3-class classifier. So very interesting question and a very good question.
Thank you. So the next question is from Ashok Narayan. So he's asking, maybe with regards to the system or the tool, does it also draw bounding box for cancerous region to help surgical or radio procedures? So he has 3 questions. So this is the first one. Yeah.
Okay. So so the thing is that this is liquid tumor. So blood cancer means that it's a liquid tumor in the sense that you're taking either the peripheral blood sample or you're taking bone marrow aspirate, but the cells are actually rotating or maybe moving in that fluid, right? So unlike the solid tumor, where you see that the tumor is localized and it's growing in nature, so even the periphery is also playing a role, the nearby regions are also playing a role, and you really need to give the boundary where the cancer tissue is or the tumor is located. So here it is not so. All the cells are independent. So for example, if I go back to the slide, so this is so this is how the slide looks like. So this is one cell. If you can see my mouse, this is another cell. So there is nothing like drawing a bounded box, but what you need to do is segment this cell out and then do the detection whether it is healthy or cancer. So I hope I have answered it. And definitely you can't do any surgery on the blood. It has to be done on a tissue. So here in this particular case of liquid tumor doesn't make sense. So then the second one is, are there any data available to determine the age of cancer cells or to know the stage of disease? So here we focused on, so again, I believe that these questions are more pertinent to solid tumors, where we, for example, for breast cancer also we talk about stage 1, stage 2, stage 3 cancer. So far, whatever work we have also worked on survival analysis of blood cancer patients. So here we talk about high risk, low risk or medium risk patients, depending on how they are, or what their survival is. But per se, we have not separately looked at the data which is corresponding to low risk, high risk or medium risk. So I am assuming that the number of cancer cells would grow large in number if it is a high risk patient and would start circulating in the entire body and start disrupting with the body metabolism, not the other way around. But definitely it's an interesting question and maybe looked at if the data is definitely prepared in that manner. So the third one is, what methods used to improve the recall in the imbalanced data? So we would say that, I don't know whether the question is related to the metric or the method otherwise. So definitely the architecture should be robust enough to handle the imbalanced nature of the data, in the sense that if you look at the folds, we intelligently made the folds such that the ratio of the cancer and healthy cells remain the same in the two classes. Now, the other way could be that we could have used the cost sensitive loss functions to take care of the imbalance in the data set. But eventually we looked at the F1 score and the balanced accuracy to see whether our performance is good or not. So particularly both weighted F1 score. So weighted F1 score and the balanced accuracy, that takes care of. So instead of accuracy, that may be a misnomer here. So we made use of the balanced accuracy and the weighted F1 score so that we know whether the results are can be relied or not.
Okay, thank you. Any other questions? Please unmute yourself and you could ask. Yeah. So I had one question. So in the second part of the work, you mentioned about the auxiliary classifier. I couldn't catch the, you say the motivation or the intuition of that channel, yeah, of that particular branch, let's say.
Yeah, so the motivation came from, again, from this part, the first work that we did. So where the idea was that if, let us say, because as I said, in the data, we just don't know which are the noisy labels. So cancer class may have some noisy labels, but we don't know. But if we are training an architecture using the noisy labels, it may happen that it's actually not learning, and over time we find that its performance is deteriorating because some samples were noisy and we are forcing it to learn it into a wrong class, right? So what we did here was, in this particular work, for example, we made use of the auxiliary classifier, where we said that only those samples in the training process which were predicted with the correct label, only those correct labels we are going to use in the auxiliary to train the auxiliary classifier. So in a way, this classifier is being trained on the data, which in our opinion, if the classifier was really trained properly, then maybe this doesn't go through, during the training process, doesn't go through the noisy labels. So to some extent, it would handle that. So that's the idea that we again extended in the next one, projection loss with sample discarding label flipping, because there could be some data set you cannot actually discard the labels, discard the samples, because there could be the one of the class may be small, you wouldn't like to do that. Medical imaging data set particularly have a problem that they are generally small in size, so you don't want to do that. So we made use of the coupling classifier again, and again you could use it as a single one, or you could use the ensemble of the decision of this as well as this in some intelligent ways that one could iterate upon.
Got it. Thank you. Any other questions? Okay.
Yes, I had one question.
Yeah,
Yes.
So there have been approaches I've seen where, for things like breast cancer, they have created synthetic data using some generative models like GANs to tackle the class imbalance. So here for blood cancer also, can such techniques be used to tackle the class imbalance?
Yeah, so very good question. So actually I spent about 1.5 years in using GANs and generating the synthetic data. But at least at that time, this was way back in 2018-19. So what we suffered with the challenge was that it was generating synthetic images that were similar to the training subjects that were used. Whereas we want performance to be really robust on the prospective subject, because every time we take it to the new subject whom we have not shown during train, val, and just want to deploy, it wasn't working. So I'm assuming that now the techniques of generating synthetic data have also improved over years compared to what it was. So maybe in the future we can think of, but particularly for multiple myeloma, if you see that we prepare data of around 80,000 images, which is not a small data set by any means. So of course, even 15,000 plus curated cell images, this is the largest data set in the public domain. As of now, we have not seen any such data set. There are hardly 280 or at max 1,000 images of BLL cancer, and multiple myeloma, I haven't seen any public data set, curated data set of this one. Yeah, there is, I think, Sonam.
Sonam, yeah.
Yeah, thank you, Professor, for a wonderful talk. So I have two questions. The first question is, when we see the cancer cell, how do we know whether the cancer has originated at that position, or whether it has been carried from another place and it has come here, right? So like whether it is a and also the second one is like let's say we have we see the cancer cell, but then it's very important for us to know, as you mentioned earlier, the stage of the cancer. So is there any way, using the same algorithm, that same process that you are doing, to find out like which stage the cancer is in?
So again, it goes to, I think the questions are largely inspired from solid tumors. So this data is for the blood cancer. So in blood cancer, it is the blood cell that becomes cancerous in nature. So, and blood, is circulating in your entire body. So you cannot say from where it is originating. It is actually the blood cells that have got malignant. So I believe the first part is answered. Am I correct?
Yeah, but then it might also be possible that there is a cancerous tissue somewhere, and through that the blood passes, and the cancer is taken up from there.
So cancer is different. You say a particular cell type becomes cancer in nature. So if it is, let us say, liver cancer, then liver tissue would become cancer in nature. Liver cells are different from blood cells. Right?
Okay,
so this is, for
morphology,
example,
morphology that can be found out.
Yeah, hold on. So for example, this particular, both these cancers, BLL as well as multiple myeloma, both of them are actually white blood cancers. But white blood also, they are different. One is because of the B type, lymphoblasts, which are got malignant. So in white blood cells also, there are a number of different types of cells in white blood cells. So it is one type, plasma cell is another type. So you have different types of cells again in white blood also. You can have BLL, you can have CLL, you can have multiple myeloma. So there are so many different types of cancer. So whenever you talk about cancer type, it is also the cell type that gets involved, and you talk about that particular cell type cancer. So it's not that if there is a liver cancer, the liver cells would start circulating in the body and they will get captured in the peripheral blood. So that's not, it's the particular cell type which is we are talking about. That would stay there. Those would be the advanced stages when they're actually the malignancy is now percolating because your metabolism of the patient has become altered, in different genes have got involved, and maybe large number of genes and metabolism has got altered. So now it is spreading or erupting in different tissues also. and that's where it is spreading, but not per se as, so this is, right now it is, we are talking about blood cancer. So it doesn't, so every cell type is different.
Yeah, thank you, Professor. I mean, I would just reframe the question. Like, is there a way to differentiate between the BA, whether it is a BLL or is it a CLL?
Yeah, that's what I said. The pathologists know that it is B-type cell which has got increased in numbers. They can immediately identify looking at the, when they look at under the microscope, this is how they give their, they sign off their report. They look at the image slide under the microscope. They can figure out what are which ones are the basophils, neutrophils, B-type cells, T-type cells, plasma cells. They can recognize different cell types, but what they cannot recognize whether a B cell is a healthy cell or a cancer cell. But they can recognize morphologically whether it is a B-type cell, eosinophil, basophil, neutrophil, or is it T-type cell, plasma cell, and so on.
What about the stages? Like, can the stage
Cancer
be
stage, I said that it is there in the solid tumor. In the liquid tumor, I haven't seen anything like that. We are working with AIIMS. If there had been something of this sort, definitely we would have looked at that, but there is nothing of that sort. It is only high risk, low risk, or medium risk staging that they do. They definitely do risk staging from the point of view of treatment or therapy in blood cancers. So you can say maybe stage 1, if you are looking at interested in staging or stage, then maybe you can say low risk stage, medium risk stage, and high risk stage. Definitely you can talk about that. But so far, what are the changes that would happen in the cells per se? Right now, it's even challenging to figure out whether it is a healthy cell or the cancer cell. If you talk about high risk, how would you figure out whether a particular cell is low risk, high risk, or medium risk? I don't see the the problem statement there.
Actually, the first question was like motivated by the presence of circulating tumor cells. So in that question, in that particular context as well, the circulating tumor cells are similar looking to the blood cells as well. So that's all right.
We have not worked on. I'm sorry, CTC we have not worked on.
Right. Okay. Thank you. Thank you so
much. But definitely, I'll look at this problem. I'll discuss with the doctor.
Thank you
so much.
Yeah.
There's one more question in the chat. Can the models created be extended to identify rarer types of leukemia, like hairy cell leukemia?
Yeah, very good question. We want to do that. So we want to actually look at, now we want to extend the work to look at different, maybe CLL and different types of cancers also. So in the near future, but again, it comes back to the same thing, preparation of the data set, annotation, curation. So that's a lot of effort. So unless that is done and a proper data set is prepared, it's hard.
Correct. Okay, any more questions? Okay.
Hi, ma'am. Ramya here.
Ramya.
Yes. Yeah, it's really a very wonderful talk, ma'am. Thanks for this enlightening us. Also, just it's a understanding the kind of like when you were showing cancer cells and normal cells, it was almost looking very similar for a man, anyone who is looking at it. So when you are in the building the architecture, in the evolution of building this right architecture, you would be touching a point, something like manually you might go and look at the cells, how to intuitively analyze this output, and all those things you might have faced. How this kind of situation you overcome it, because sometimes it will be like a monotonic, disgusting or frustrating, something it could happen. How this kind of situation?
So sorry, I am not able to follow the question. Is your question related to that if they're looking similar? How
yes.
Annotate the GT?
It's annotation is anyway there, but when we look at some architecture and its output, we may not able to identify why this is really happening, because manually we couldn't analyze like what is right and what is wrong. So in this kind of situation, what is the motivation factor for you, and what kept you really in what do you call, in the same shoes to something like that, right?
Okay,
This kind of
I think that I don't know if I have followed the question correctly. So is that you're saying that if they look similar, then how confident you were in making the problem statement, whether it is going to work out or not?
yeah, when you analyze the results, right, what are all the approaches you take? Because
Okay.
it is when you look at F1 score or any kind of parameters, it is a number again. When you go anytime, because it's an image, right, you naturally tend to like go and look at the image. Is there any visual clue which I can make out
Yeah,
what
good
happened?
question. Yeah, good question. So I think that Ramya, you're touching on the interpretability of the method.
Yeah, yes.
So yes, so definitely we should be seeing it more carefully. But as I explained in the beginning, that we made the architecture looking at how the images are captured, or where the image would have the difference. For example, I talked about the motivation for building the SD layer architecture. We all know that CNNs are being used, but we looked at where the difference would be. So in our opinion, the difference would have been there in the texture. But what texture? Is it the RGB, or is it the staining chemical, which is what is being absorbed depending on the intracellular structures? So that's where we looked at the problem statement, and we came up with the front end layer, the SD layer, the idea of SD layer. Then we again drew inspiration that flow cytometry uses spectroscopy to differentiate in the cell type. So should we be using some frequency descriptor somewhere? And we appended a DCT layer, and we looked at whether it is working fine. Now, rest of the thing are actually still that we have built a CNN architecture, and we are looking at how this CNN architecture is doing. But definitely this particular problem statement requires another level of work where interpretability analysis should also be done, and whether that interpretability analysis can be linked with the intracellular granularity that might be known depending on the bio of the problem. Perhaps it would be really a wonderful work. So very good question. I would say yes, definitely we should also be doing it.
It's almost like more and more you will be knowing more about the domain also. Exactly. To understand the problem, right?
Exactly.
Yeah.
Definitely.
Thank you.
Thank you so much for asking this question. Wonderful question. Thank you.
Thank you. Thank you.
Okay, I think there's good participation, lots of interesting questions, and thank you for keeping it very interactive and a wonderful talk. So I think it's probably good time to conclude. Thank you very much, Professor Anubha, and maybe next time we'll invite you here physically. Please do visit our campus. And over to you, Nikhil. Yeah, thank you very much.
Thank you so much. Thank you so much for inviting me and giving me this opportunity to share my work with you all. Thank you so much.
Thank you, Professor.
Thank you.
Once again. And also thanks to Professor Dinesh for sharing the session.